Okay, so today we're looking at cyclin, a protein specified by maternal mRNA in sea urchin eggs that is destroyed at each cleavage division. This was done by Tom Evans, and it was published in 1983. So the objective of his experiment is he wanted to determine how this newly discovered protein, which is going to later call cyclin, was incorporated during the cleavage process of embryos, and cleavage is just simply the division of the embryo. So it's pretty much just like mitosis, essentially. So the background information here, so first of all, they had studied an organism that they call Arabia punctulate, and it's just a sea urchin is what it is. And so in this cleavage process, it has 8 rapid divisions that require continual protein synthesis to sustain it, which is which sounds very normal. It's a very normal thing that happens when cells are dividing. And they had essentially determined that there was 3 or 4 proteins that were coded for by maternal mRNA. They obviously don't go into how they knew this, but they had said that there was these 3 or 4 proteins coded for by maternal mRNA, and that each time they were essentially they had seen or observed that each time they would divide, that one of the proteins is destroyed, which we're going to see later on in cyclin. Then also the sort of general thing here is that fertilization of eggs comes with an increase of protein synthesis, as well as a change in the pattern of synthesis, which that more so is going to apply to cyclin, as we're going to see later on. And obviously cyclin is observed, which I kind of just said here, but cyclin is observed to have a cyclic pattern in its concentration after fertilization starts. So it's not really present at all before fertilization, but after fertilization, it's present. And as we're going to see later on in some of the results here, it's going to go up and down, up and down. And then, so a side note here for help to explain the results later on is in sea urchin eggs, protein synthesis can be activated by parthogenic compounds like NH4Cl and A23187. So essentially what these compounds do is they pretty much start parthenogenesis, which is pretty much kind of like an asexual form of reproduction, sort of. But it mainly only really allows for proteins to be synthesized, and it does not really allow for the whole process of the cell division to actually occur. It's more so just on the proteins to be synthesized. So the methods. So I'm not going to go very in depth here with their methods, because if I were, it would take all day and we would never get to the main important things. So for clarity, I'm omitting most of the methods here. But what they had to do in general is they obviously had to prepare their samples. Then they had to, another side note here, they had to prepare and incubate their eggs and embryos, so obviously their samples were ready for their experiments. And then some of their experiments involved, obviously, is fertilization and labeling, obviously, so they can track cyclin. Then for their analyses of samples, so they did a lot of gels. They have a graph as well we're going to see later on. And one thing here that we'll explain is the cleavage index, because the cleavage index is important later on, and that's just essentially just a measure. It's just counting the number of cells that have divided from one step to the next. So that's all that is. So, and now in the results section. So this is the first main figure here. So this figure is demonstrating the different levels of protein synthesis between an unfertilized embryo, fertilized, one's been activated with A23187, and one that's been activated by NH4Cl. So first of all, the numbers on the very far left, the 11668, those just represent mass. Those are not as big of a concern here. What is important though is, so the lanes A through J all represent a difference of 10 minutes, and A starts 25 minutes after the embryos were incubated. So A is 25 minutes and J goes all the way up to 115 minutes, and that stays true for all of them. And then K represents 127 minutes later on, except for UK. Well, UK and FK in the very, very far one on the right represents 127 minutes as well, but I'll get to that a bit later on. And then, so the important things here though is to look at the unfertilized gel, and you see X, Y, and Z. And these just represent polypeptides that has their synthesis adjusted and changed after the activation of fertilization, or either the 2 compounds represent. Because as you can see, if you look, X, Y, and Z, the bands are pretty much non-existent. If you go from the left to the right, they're pretty much not there, because these are compounds that their synthesis stops after fertilization happens. So the proteins that we are concerned with, though, is where you see A, B, and C. And essentially, so protein A is cyclin, so I'm just going to refer to it as cyclin from now on. And B and C are other proteins. I don't give them names, they're just B and C. So cyclin, or otherwise protein A, in the fertilized gel, what we're supposed to see here is that, so from C to G, they're saying that the band is sort of, it kind of initially starts to increase and becomes more intense, but then by the time it gets to G, that the band is pretty much gone. But then the band reemerges again and becomes intense from H to J. And of course, you can still see a little bit in K. And which is essentially what that's trying to show you is that cyclin goes up and down in its concentration, which is the important thing of cyclin, which is why cyclin eventually gets its name of cyclin. Then the main difference here is between the fertilized and the ones that were treated earlier with NH4Cl and A23187 is that in those two, what they're saying is you don't actually see the cyclic nature of cyclin. And that's because, and essentially what they conclude from this is that means that there must be some sort of like essentially the natural process of fertilization, something with cell division there must be important to make cyclin actually be destroyed, because it's not being destroyed in those two, because otherwise if it was, you would see fainter bands, or obviously they're not as essentially intense. And so the last part here is UK and FK, which are again on the very far right of the right hand gel. And so U stands for unfertilized and F is fertilized. And these are again are at 127 minutes after the incubation. And essentially what they're showing, those are what they call a light sort of, like it was like a light treatment with it essentially. So and those are just simply there for comparison to the K of the NH4Cl. But so it doesn't really elaborate much about again comparison, but that's why those are there. Okay, so their next results, or I should say their next important figures here are these three. So then we'll start at the left-hand side with the graph. So the graph is essentially, what the graph is showing, the graph is essentially confirming what figure 1 was showing, what the protein synthesis was showing, and you can just ignore the gel at the top. I'm not sure what that, they don't explain what that gel is trying to show, so I'll just ignore that gel. So, okay, so first of all, so the dotted line that goes up and down at the bottom there, or the square line, that represents the cleavage index, which again is just a count of the number of cells that have actually divided, which makes sense from 0 to 1. Those are in hours, and you're not having a lot of cells dividing, and then you're going up, and then you're going, obviously that makes sense for the cell cycle purposes anyways. Another thing is like they had a bunch of, it's a sample of a bunch of different embryos, obviously, so that's why, that's why they have a lot of, obviously, cells, right? So, okay, so then they have in A, so they have A and B is shown. C is not shown for clarity purposes, but C would look pretty much exactly like B, but C, for clarity purposes, C is omitted here. But so A, as you can see, is the cyclin graph, which is going up. So as you can see, actually, cyclin is being produced after fertilization, but the first division actually hasn't happened yet, which is trying to show you that cyclin needs to be produced in the first phase before division can actually happen. But then once the first division does happen, then you see the concentration goes down substantially, which is trying to show you that cyclin is being destroyed. And then it starts to go back up, and then it gets destroyed again. And then we see the whole sort of cyclic process with it, which goes hand in hand with the cell cycle. And they've essentially done this by performing like a density test, essentially to try to do, which allowed them to determine like sort of like the density, how much was actually there throughout the whole process. So that's how they had produced these graphs. And B essentially is just following the normal sort of protein like linear progressions you would normally see. So, okay, now we're going to go on to the middle one. So the middle gel is an extension of a previous experiment. I'm not showing it here. I'm not showing the gel from that because I didn't really feel that was important to help represent what they're trying to show you here. So the middle one, though, they had a. So first of all, the first one starts off with 40 minutes after essentially the previous experiment, which I was trying to, which I'm again not showing here. And obviously, let me just quickly say, so obviously A, B, and C all correspond to the same things. Obviously, B and C are the proteins, and A is cyclin. And let me quickly go back one step further, is that the reason why what this gel is trying to show you is trying to show that cyclin is still being produced after the third stage, because due to problems they had before, is that they unfortunately couldn't determine that cyclin was being destroyed after the third stage. They lost track of the synchronousness of it essentially was gone. So in order to determine that it was still being produced, what they did is they're using a compound called emetine, which essentially blocks protein synthesis. So now let me get back to what I was saying earlier. So one is 40 minutes after it was a sample taken 40 minutes after the initial sample from the previous experiment. Then 2 was taken 3 hours after, and 3 was taken 3 hours after, but it was treated with emetine. And as you can see, the cyclin band completely disappears, which is evidence that cyclin is still present there, or that cyclin is still being made, but then obviously it's gone, right? So 4 was taken 5 hours after, which you can still see the band, and 5 and 6 were both taken 10 hours after. And the bands, they were saying that the bands are still visible, barely visible, and even though they're not showing it here, but they had also said that once 5 and 6 had been treated with emetine, that those bands also disappeared, which is more, again, evidence that even, say, 5-10 hours after the division had started, that cyclin is still being produced and then going away, which is, again, also trying to show you that the cyclin has this cyclic pattern. And the very last graph, the figure they have here, is so cytochalasin and cholecine are essentially two things that they stop the cell from dividing, but they prevent cell division. That's what they do. And so an A in both cases represents it's a control, right? It represents the embryo is untreated with these drugs. And B, it represents them when it's treated with the drugs, or with their drugs. And essentially the sort of like, obviously the lines you're sort of seeing in each band is represented of the cyclin levels they're trying to show you here. And so essentially what you're seeing here is that, so in A, it proceeds as normal and cyclin is pretty much broken down and it's pretty much all gone as it progresses. But then with the cytochalasin and the cholecine that they were showing here, that these drugs are actually slowing down the degradation of cyclin. And by the time you get to 12, which is their 12th sample that they had here, is that it's still there, that these drugs actually can modify cyclin, the way that cyclin is degraded. So this actually is proof that, again, that cyclin still has its fluctuation patterns from before, and it can actually even be modified by different other sort of drugs. So, what does all of this sort of mean? Well, they go through a bunch of different things when they're discussing, so I'll just kind of hit on a few of the points. So, most of the proteins, they follow a linear increase in concentration after fertilization has begun, which does make sense because obviously as the cells grow, obviously you need more and more protein to obviously compact with that growth. Then that cyclin, as we saw from the results earlier, that cyclin, on the other hand, it starts linear, but then it's destroyed and will build back up again and again. And they said that this is a very reproducible thing that they were observing, which is a very important thing with cyclin. And then, so this probably seems like a very weird point that they bring up. But so if you remember earlier in the test, all about maternal mRNA, and so they had done some testings and things like that, because previously thought before they discovered cyclin is that mRNAs had mainly coded for tubulin because they've always seen that it had associates with like the spindle poles and things like that. But after they discovered cyclin, they did testing from this because they knew that it was coded for by maternal mRNA. And what they did is they tried to bind it with tubulin antibodies, and they could not get it to bind, which told them that that's not the case, that maternal mRNAs can code for other things besides just tubulin. So that was essentially what they were doing, is they were confirming that cyclin was not part of the tubulin superfamily. And then they finally get to some of the more interesting things to talk about cyclin. So they had believed that cyclin had been linked to the cell division they had said that this experiment did not have any sort of direct evidence that it is, but after obviously looking at all of everything they had done, they had strongly believed that they thought that would be almost impossible for cyclin, for the way cyclin functions to not be a part, or to not somehow be linked to the cell division. And then they had sort of tried to speculate of what cyclin's role is in cell division. And they had believed at the time, anyways, from some of the other experiments that had been done, is they had believed that cyclin probably had a role in breaking down the nuclear envelope, which we now know is wrong, because we know that cyclin is a part of the CDK family, or the cyclin-dependent kinases. So we know that cyclin plays a role in binding to the CDK, and then which then changes its shape to allow it to be phosphorylated and dephosphorylated, so the CDK can actually be fully functional. Other we know that kinase is obviously going to phosphorylate other things. So that's what cyclin does. It just simply is involved in activating the CDKs. And this, which obviously we also know that play a major role in regulating the cell cycle. And so essentially the conclusion they have from all of this is that they're like almost convinced that the cyclin is somehow involved in the cell cycle. Again, they didn't have evidence of this, but they were like almost 100% sure that cyclin had to be involved in the cell cycle, and that it follows a different pattern of production than most of the proteins do, because, as I mentioned earlier, cyclin has a fluctuation pattern where it goes up and then down, up and down again, and it's reproducible, whereas most proteins will just have a linear progression where they go up, 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 and up, and they don't actually ever go back down.